Hello everyone, I'm myself Dr. Dipti Kariya and today we'll discuss about white blood cells also known as leukocytes. Before starting my class, I would like to inform you all that you will get all my lecture notes in soft copy and hard copy. To get the soft copy, you have to download the application Vesalius and you have to get the course. And for hard copy, you can order my book that is Physiology Notes which is available on Flipkart and Amazon. Link for the book as well as link for the application, they all are given in the description box. So let us start with today's topic that is white blood cells. Okay. White blood cells, which are also known as leukocytes, these are colorless nucleated elements of the blood and they play an important role in the defense mechanism. Important function of WBC is they provide defense. Okay, this one. Now, what is the size of WBC? Size of WBC that is around double than RBC. Diameter is about 10 maximum, minimum 10 to 14 micrometer diameter. Okay, they are nucleated cells. They are the only cells in the blood having nucleus, and they also have granules. And according to presence or absence of granules, these are granulocytes and agranulocytes. We will discuss in detail. Normal count of WBC that is about 4,000 to 11,000 cells per cubic millimeter of blood, and lifespan depends on the defensive action of the body. Now, so you can see here, depending on the presence or absence of granules, this WBCs or leukocytes, they are divided in granulocytes and agranulocytes. You can see here, granulocytes are further divided in neutrophils, eosinophils and basophils. These are the normal percentage of all this. Neutrophils are 60 to 70%, eosinophils are 1 to 4%, and basophils are 0 to 1%. Lymphocytes are 20 to 30% and monocytes are 2 to 8%. So here you can see this we have already discussed. Normal count in adult that is 4,000 to 11,000 per cubic millimeter. At birth the count is highest. This is about 10,000 to 25,000 cells per cubic millimeter. This is normal physiological leukocytosis. Okay, up to 1 year of age the count again remains high, 6,000 to 16,000. Okay, and these are the values of differential count. Okay, differential count that is in percentage means here we are counting 100 cells. From this 100 cells, 40 to 70 are neutrophils. But if suppose we are counting 4,000 cells total, then we will get the absolute count, and absolute count is this is the value. How can we get this absolute count? You can get by the formula. You can say the suppose from 100 cell, suppose I 100 cells I found 45 neutrophils. Okay, and total count that is around 5,000. So how many neutrophils? And you will get the absolute count. Fine. Now, variations in WBCs, leukocytosis. What is leukocytosis? That is increase in the total WBC count above normal value. Normal value, as we have discussed, 4,000 to 11,000. So when it is above 11,000, that is known as leukocytosis. Okay. What are the physiological causes of leukocytosis? We have already discussed. Physiological causes are age. In newborn baby, it is high. During exercise and emotional condition, they are again high. Mental stress, again the count is high. Food intake also increases the count. Diurnal variation, during daytime also in the evening it is high. Pregnancy, count is high. Parturition, the delivery of baby, also the count is high. Sleeping, the count is reduced, and during menstruation the count is high. Now, pathological variations are, what are the pathological causes of leukocytosis increase in the WBC count? They are, number one is acute bacterial infection. Pyogenic means pus forming organisms, they increase WBC count. Acute hemorrhage also increases WBC count. You can see here, this is high count. You can see here, this is the smear showing high count. In allergic conditions, particularly eosinophil and basophils are increased. In burns also the count increases. After operation, post-operative period, common cold, viral infection, tuberculosis also increases the count. Other, that is leukopenia. Leukopenia, that is decrease in the WBC count when the count is less than 4,000 per cubic millimeter of blood, less than 4,000. Okay. What are the causes for leukopenia? That is infection by non-pyogenic organism. Commonly typhoid fever, paratyphoid fever shows leukopenia. Viral infection also shows leukopenia. Protozoal parasitic infection like malaria, it shows leukopenia. Starvation, lack of food, malnutrition, count decreases. In case of bone marrow aplasia, when bone marrow decreases or bone marrow is not performing its function, then also the count decreases. Certain drugs, they also produce bone marrow aplasia. Okay, chloramphenicol and certain other drugs. Repeated X-ray exposure, repeated radiation, okay, also produces, and certain chemical poisons like lead, arsenic, they also produce leukopenia. Next is leukemia. Leukemia, what is leukemia? What is the difference between leukocytosis and leukemia? You can see here, this is the blood picture of leukemia. So what is leukemia? That is, leukemia is malignant, it is malignant disease of blood, malignant disease of blood in which there is increase in the total WBC count, okay, with immature WBC. Why immature? Because there is malignant proliferation of either myeloid or lymphoid series. Myeloid series that produces granulocytes and lymphoid series that produces lymphocytes. Okay, so this is leukemia, malignant transformation of WBCs because of the uh, change in the series. Okay, that is malignant transformation. So WBCs produced, they are immature. You can see here the cells are immature here. Okay, these are mature cells, and the count is, most of the time it is more than 1 lakh per cubic millimeter. And malignant proliferation of WBC precursors that results in immature cells, immature leukemic blast cells are produced. Okay, and because of bone marrow failure to produce mature cells, mature cell production decreases, so the person is having anemia, decreased RBC, leukopenia. Leukopenia means normal WBCs are reduced, okay, and thrombocytopenia, thrombocytes are reduced, thrombocytopenia. Okay, immature WBCs are increased, but mature WBCs are decreased. Okay, so this is the characteristic of leukemia. Now, what are the types of leukemia? There are 4 types. You can see acute and chronic, for they are mainly divided, and this acute and chronic are again divided in acute lymphoblastic and myelogenous. So acute lymphoblastic leukemia, acute myelogenous, chronic lymphoblastic and chronic myelogenous. Myelogenous series, when there is proliferation, then myelogenous, and lymphoblastic series proliferation that results in lymphoblastic leukemia. Okay, now Now, what are the causes for leukemia? Genetic causes are there, genetic factors for the leukemia is also one of the cause. Certain chemicals like benzene, benzene, and certain cytotoxic drugs, they also produce leukemia. Viral infection also sometimes they produce leukemia, and ionizing radiation, if continuously and repeatedly exposed, that results in damage to the DNA of the cells, and that results in malignant transformation of the cell. Now, lifespan of WBCs. Different WBCs have different lifespan. Neutrophils lifespan, that is about 2 to 5 days. Eosinophils 7 to 12 days. You have to remember all these values. Basophils 12 to 15 days. Monocyte 2 to 5 days. And lymphocytes half to 1 day. Okay, now we discuss about leucopoiesis. Poiesis means production. So origin, development and maturation of leukocytes. Okay, these leukocytes, as we have discussed, RBCs, WBCs and platelets, they all are produced from the stem cell, and these stem cells are known as PHSC. We have discussed with erythropoiesis. You can go and watch that video that I have given this name. Full form of this PHSC is pluripotent hemopoietic stem cell. Means all the blood cells are produced from this cells. Pluripotent hemopoietic stem cells. Okay, from which RBCs, WBCs, and platelets, they all are produced. And this granulocytes, they develop mainly and exclusively in the red bone marrow. So that is known as extravascular leucopoiesis. Okay. So here, this pluripotent hemopoietic stem cells, they are differentiated to form colony forming unit, and colony forming units are for granulocytes and for lymphocytes. Okay. So this colony forming unit for granulocytes, they produce granulocytes having granules, neutrophil, eosinophil, basophils, and colony forming unit for lymphocytes, they produce lymphocytes. Okay, so you can see here. This is pluripotent cell. You can see here in this diagram. Then colony forming unit, granulocytes. You can see here. Okay, and monocyte. These are granulocytes. Basophil, eosinophil, and neutrophil. And monocytes are also produced from the same series. Colony forming unit, monocyte. Okay, and lymphoid stem cells from which lymphocytes are produced. So this is the basic steps. Okay, now myeloid series. In myeloid series, the cells are, you can see here, immature cells are always known as blast. You can see here in this. So myeloblast, then they are converted into promyelocyte, then myelocyte, metamyelocyte, then band form. You can see here all these pictures, all the cells are shown in the picture, and band form as we have discussed, and ultimately granulocyte is produced. Okay, duration for this myeloid series granulopoiesis, that is about 12 days. Okay, erythropoiesis, that is 5 days, 7 days, 5 days for the production of reticulocytes, and two days are required for conversion of reticulocytes to erythrocytes. Okay, and these granulocytes, they are stored in the bone marrow, and numbers of stored granulocytes, that is 3 times more than the granulocytes present in the circulation. Now we'll discuss morphology of each and every cell. We have discussed the series. You can see here this myeloblast, promyelocyte, myelocyte, metamyelocyte, band form, and granulocyte. So let us start with myeloblast. Blast is always immature cell. Size is 16. Always immature cells, they are large in size. So 16 to 20 micrometer diameter. Nucleus is large. You can see here in this diagram, large, round to oval, purple blue in color, and it contains, you can see here, chromatin, fine chromatin, and it contains 2 to 5 well defined pale nucleoli also. These are the nucleoli and these are the chromatin, they are present in the nucleus. Mitosis is marked +3, okay, in myeloblast. Next stage, that is after myeloblast, that is your promyelocyte. Size decreases. You can see here, this is promyelocyte in this diagram. Size decreases. Here this becomes 14 to 18 micrometer diameter. Nucleus, that is round to oval. You can see here in this diagram, slightly small and fine and condensed chromatin. Now chromatin gets condensed and less prominent, and few nucleoli are present, less prominent as compared to myeloblast. And cytoplasm, here cytoplasm amount increases. Now nucleus decreases size in size, and there is orophilic granules, primary non-specific granules, they give positive reaction with the peroxidase chain. Mitosis, again here it is marked +3. Next stage, that is myelocyte. Size again decreases, 12 to 16 micrometer. Nucleus, now this is here, you can see myelocyte, this one, this is myelocyte. Nucleus is eccentric in the corner, not in the center. Okay, round to oval shape, round to oval, eccentric. Okay, and it has got coarse nuclear chromatin. You can see here inside there is chromatin. Cytoplasm, specific secondary granules are present, and accordingly the cell can be identified as neutrophil, eosinophil, basophil, myeloblast. Neutrophil, myeloblast stained by both the dyes, eosinophil by red color and basophil by blue color. Now primary granules are also present, but their formation is now stopped. Mitosis is continue here. Next stage, that is metamyelocyte. Here again size decreases. Now it is near to the mature neutrophil, eosinophil, basophil, 10 to 14 micrometer diameter. Nucleus decrease in the size, become indented. You can see here indentation is there, depression slightly. Nuclear chromatin is dense and clumped, and nucleoli are absent now. Cytoplasm, amount of cytoplasm increases, it becomes more liquid, and primary and secondary granules are present, and according to the nature of the secondary granules, this cell is classified either as neutrophil, eosinophil, or basophil. And granules, they show amoeboid movements. And this amoeboid movement that is maximum seen in the neutrophils. Mitosis now stops at this stage. At metamyelocyte stage, mitosis now stops. Next, that is band form. You can see here in this diagram, this is band form. Size slightly smaller than metamyelocyte, nucleus further condensation, okay, and it becomes band shaped. You can see here. Cytoplasm is pink and it contains 5 evenly distributed granules, and it has now mitosis is now absent, as we have already discussed. Okay, cytoplasm has fine evenly distributed granules present. Next is monocyte macrophage series. Now here in monocyte series, we have, you can see here, this we have discussed about myeloblast. This myeloblast, they produce neutrophil, eosinophil, basophil. Now what about monocyte? For that we have this series having monoblast first. Then this monoblast matures to form promonocyte, and from promonocyte, monocyte is formed. Okay, so monoblast, that is larger, similar structure to myeloblast. Here these two have similar structure, myeloblast and monoblast. Myeloblast for myelocyte and monoblast for monocyte. Okay, promonocyte, that is young monocyte, and size is slightly larger, 20 micrometer diameter. Large and indented nucleus is there. Nuclear chromatin is arranged in loose network. Cytoplasm is basophilic. Orophilic granules are present, and they are larger than normal monocyte. Again, you can see here in this diagram, this is the nucleus, cytoplasm. Okay, now, next is monocyte. Now it gets mature to form monocyte. This is your mature monocyte. This mature monocyte migrate to spleen and lymphoid tissue and transformed into tissue macrophages. Okay, monocyte, if it is there in the blood, that is known as monocyte. It has got, we all know the morphology of monocyte. It has got kidney-shaped nucleus. Okay, and it has got this slightly eccentric, and granules are orophilic. Fine granules are present, otherwise they are usually not seen by the normal microscope. And when this monocyte migrates to tissue, it forms tissue macrophage, and it forms a part of tissue macrophage system, or it is also known as reticulo endothelial system, endothelial or monocyte macrophage system. Both are same. This is the synonym of monocyte macrophage system. Last one, that is lymphocyte. Lymphocyte formation, as we have already discussed, all the cells they are derived from pluripotent hemopoietic stem cells. So lymphocytes are also produced. Then from this pluripotent hemopoietic stem cells, there is formation of lymphoid stem cells, and this lymphoid stem cells, they migrate to thymus and peripheral lymphoid tissue, and they are proliferated and they get matured to form mature lymphocytes. Bone marrow and thymus, they are primary lymphoid tissue, or you can say lymphopoietic organs, from which lymphopoiesis means lymphocyte production takes place. And lymphoid stem cells, they undergo spontaneous division in this thymus and bone marrow. Now, these are primary. Primary are, as we have discussed, bone marrow and thymus. And secondary, or you can say reactive lymphoid tissues, are our lymph nodes, spleen, and gut associated lymphoid tissue. These are secondary lymphoid tissue. Why are they known as secondary lymphoid tissues? These are the tissues which actively produce lymphocytes from the germinal centers of lymphoid follicle as a response of antigenic stimulus. Means when antigen comes and antigenic stimulus comes, then these tissues, they produce lymphocytes from the germinal centers of their lymphoid follicles. So this is about lymphocyte. Now lymphoid series, as we have already discussed, lymphoblast are earliest recognizable cell, and this is actively dividing cell. It resembles myeloblast. Okay, which are how it differs from myeloblast? Here only a difference here is nuclear chromatin is slightly clumped and stippled as compared to fine meshwork in the myeloblast. In myeloblast we have fine meshwork of chromatin, and here we have slightly clumped and stippled chromatin. Nuclear membrane in lymphoblast is fairly dense as compared to very fine membrane of the myeloblast. Okay, in myeloblast membrane is fine, here the membrane is dense. Okay, Then after lymphoblast, we have pro-lymphocyte. That is intermediate state. Diameter is 9 to 18 micrometer and nucleus is round, indented. You can see here with slightly stippled or coarse chromatin having 0 to 1 nucleoli. And cytoplasm is scanty and granules are absent. Lymphocyte, that is a mature cell. Lymphocytes, they are pro-lymphocytes which are converted into lymphocytes. This pro-lymphocytes, they get mature and they form lymphocytes. Lymphocytes are large and small lymphocyte according to maturity. If the cell is immature, that is large, and if it gets mature, that is small lymphocytes. Some lymphocytes enter the thymus where they are processed and they form T lymphocytes. In thymus, there is a factor, that is hormone, that is thymosin, which play an important role in the maturation of T lymphocytes. Some lymphocytes, they are processed in the liver and bone marrow and they form B lymphocytes. So T and B lymphocytes, they are according to their function. Small and large lymphocyte, that is according to their morphology or according to their age. If the cell is having less age, or you can see young cell, that is larger one, large lymphocyte, and if the cell is older, that is small lymphocyte. Now, regulation of leucopoiesis. There are certain cytokines, they regulate leucopoiesis. Cytokines include colony stimulating factors. What is colony stimulating factor? These are the glycoproteins. They are formed by monocytes and T lymphocytes. Okay, colony stimulated, and they stimulate certain colonies, so they are known as colony stimulating factors, and they are granulocytes, monocyte, colony stimulating factor. And certain cytokines, they form interleukin by monocytes, macrophages and endothelial cells. Okay, epithelial cells. Cyto means cells. Cells, they produce certain cytokines. Okay, and prostaglandins also, they play an important role in leucopoiesis. Prostaglandins are produced by monocyte and lactoferritin. Okay, so they play an important role in leucopoiesis. So this is all about leucopoiesis. Here we are ending our class. Again, I would like to inform that you will get all my lecture notes, whatever I have taught you. For that, you can get it in soft copy and hard copy also. For soft copy, you have to download the application Basophils and you have to get the course. And for hard copy, you can get my book. You can order my book, that is available on Flipkart and Amazon, and link for all that is given in the description box. So thank you so much. If you like my video, if you like it, you can share with all your friends and subscribe my channel. Thank you.